I would like to introduce with a couple of words the Biological Research Barrier at the Biological Research Center. This group has a long history because one of the founders of this group, the late Professor Ferenc Jó, he already started experimental work on blood-brain barrier more than 40 years ago. We have many research topics. Here I would like to show you the two major research areas we are active. One is the blood-brain barrier damage and protection in diseases, and we especially focus on Alzheimer's disease, epilepsy, and together with the pancreas research group of the university, we also study pancreatitis, but we study diabetes, and with Hungarian and French groups, ionizing radiation studies we also perform. A second major area is the drug delivery across barriers in general, not only blood-brain barrier, but we model respiratory barriers like the nasal, the lung barrier, and also we have experiments on intestinal barriers. That's extremely important for drug delivery, especially oral and nasal delivery of drugs, and there is a whole new area of nasal delivery. We try several means how we can more effectively deliver drugs across these biological barriers. One is the modulation of intercellular junctions with peptides and lipid molecules, which is tremendously interesting. And another one, which is highlighted here, is the use of nanoparticles, and especially targeted nanoparticles. So my student Dóra will join this project, and she will talk about that a bit later. We have several types of models and techniques. I just would like to show you here that we use transval models, and we are modeling our barriers not only with monocultures but with double or triple co-cultures, which is really unique. And I would say we are this is rather a state-of-art method, and not many laboratories can do that. And we even further developed that model with new microfluidic chips. And this is, I think, really at the edge of our present knowledge. Our techniques are mainly cell culture techniques, so we use primary cells and cell lines. We have a whole new area of techniques on especially cultured cells, like viability assay, resistance impedance monitoring, permeability measurements, classic assays like protein and nucleic acid assays, and also we do quite a lot of microscopy, immunohistochemistry, confocal microscopy. And finally, we also do in vivo measurement of permeability to validate our in vitro findings on in vivo models. We have basically in many countries in the world cooperation partners. We are very proud. We have a long cooperation with Japan, especially at the University of Nagasaki and Fukuoka. But we have active bilateral cooperation with Mexico and also with France. There is new cooperation with Norway, and I hope that several of my students participate in cooperative research visits, and I hope that Dóra will also participate in these. These are the present members of my team. You can see that there are many students, especially MSc and BSc students, not only students of biology, but students who learn bionics. And I'm really proud that we have pharmacy and medical students too. So there is a variety of background in my team, which is very, very useful, and the students exchange a lot of ideas with having different backgrounds. And now I would like to introduce Dóra Hantosi, who is who entered. Oh, this is Hungarian. I'm sorry, I didn't recognize. So she is a first-year medical student, and she has also entered this program of Szeged Talented Mentorship. She finished her studies in the Radnóti Miklós Secondary School, where she learned the biology from Victoria Gall. This is important because I really would like to recognize the work of our teacher partners at secondary schools. And she participated on several competitions during her years and got really good prizes, which I'm very proud of. And especially she had, as a secondary student, she had already a research visit in Budapest at the Center for Natural Sciences, where she already studied nanoparticle production and also testing. And that was one of the reasons why she joined my team, where she could continue this work. And with that, I would like to ask Dóra to present her talk. Thank you for your attention.
Dear chairman, dear mentors, dear teachers, and dear fellow students, I would like to summarize my work as a first-year senior student, which I did in the laboratory of Mária Deli in the Biological Barriers Research Group in the Biological Research Center in Szeged. I joined the project which is studying nanoparticles which were designed for drug delivery, and we examined them on brain endothelial cell culture models. But first I would like to say a few words about what the blood-brain barrier actually is. The blood-brain barrier is a dynamic interface which consists of the capillaries of the brain and creates the homeostasis for the brain function and acts defensively against toxic agents in our body, and provides also the brain with the needed nutrients such as amino acids or, for example, glucose. And it also plays a crucial part in the communication between the periphery and the central nervous system. But there are also problems with the blood-brain barrier in case of drug delivery. And what are these problems? The first major problem is that the blood-brain barrier prevents the majority of the uptake of new drug candidates, which are designed for treating the diseases of the central nervous system, such as Alzheimer's disease. And also there are small lipophilic molecules like classical antihistamines, which can cross the BBB and get into the brain, causing several unwanted central side effects like sleepiness or dizziness. So the question is, how can the BBB block the entry to the CNS? There are several blocked pathways on the surface of the blood-brain barrier, but I would like to introduce the three main ones. The first one is the paracellular traffic, which is blocked by tight junctions, and these tight junctions are preventing the brain from uptaking the water-soluble molecules. The second blocked pathway is the pathway of the efflux transporters, which are protecting against the xenobiotics and drugs. The third blocked pathway is the blockage of the non-specific vesicular transcytosis, which ensures us that there is no plasma protein leakage at the blood-brain barrier. And there are, of course, open pathways which we can use for drug delivery. The first main one is the pathway of lipid-mediated free diffusion, which allows special drugs and, for example, drugs of abuse and small lipid-soluble molecules to cross the BBB. The second pathway is the pathway of the solute carriers, which are providing the brain with the required nutrients like glucose, amino acids, lactate, or vitamins. The third pathway is the pathway of receptor-mediated transcytosis, which consists of three main steps. The first main step is the receptor-mediated endocytosis on the luminal side of the brain. The second step is the transcytosis through the very thin layer of the brain endothelial cells. And the third step is the exocytosis on the brain side. This transcytosis carries peptides or proteins such as insulin or transferrin to the central nervous system. And what are the strategies that we have to deliver drugs to the CNS? For example, the first main strategy is to circumvent the BBB, of course with using the direct delivery of the drugs or using the nasal pathway. There are also usage of modificator molecules, for example small peptides which can open the tight junctions reversibility, as you can see here. And there are a third way, which is targeting the BBB transport pathways, which I mentioned before, by modifying our drug molecules or by using targeted nanoparticles, which we used in our experiments. During our research, we used an in vitro model, which consisted of brain endothelial cells. As you can see in these pictures, which are an electron microscopy and an image staining picture, that our model had the characteristics of the in vivo BBB, such as the tight junctions and the caveoles used for transport. In the second picture, you can see the tight junction proteins, such as claudin 5, beta-catenin, and ZO1 image stained. We used targeted nanoparticles and niasomes for our experiments. Our niasomes were a diameter of 100 nanometer and filled with an Evans blue albumin complex as a cargo. We made uptake and transfer tests, and we wanted to test the drug delivery by the glucose transporters on the brain endothelial cell's surface. So to use these transporters, we targeted our nanovesicles with glucose analogues. As you can see on the graphs, the glucose analogues increased both the uptake and the transfer of our nanovesicles. We also used solid nanoparticles, which were made from a fluorescent red polystyrene and were labeled with neutroidium. We wanted to use the glutathione and the transporter and the multivitamin transporter, which are highly expressed on the surface of the endothelial cells in the blood-brain barrier. So we targeted our particles with biotin, which you can see here on this picture, and the biotinylated glutathione, which you can see here. We did uptake tests, and as the graphs and the picture shows us that the particles which were targeted, so in the case of the particles which were targeted, the uptake was increased. As a summary, I would like to emphasize that the blood-brain barrier is crucial for the nutrient transport of the brain, but it can also act as a blockage for drug delivery. But it is important that we can exploit this blockage by using special techniques, such as our targeted nanoparticles, which can cross the blood-brain barrier, so can be used as for drug delivery. We also have plans for the future, such as introducing new drug-like cargo, curcumin, to our nanovesicles, and the usage of glutathione-targeted nanovesicles. I would like to say a special thanks to my mentor and my co-mentor, Mária, Deli, and Silvia Veselka, and I would like to also thank the support of the Foundation and the University of Szeged. Thank you for your attention.